This minute biophysics video is about hydrogen-deuterium exchange mass spectrometry. Within our body, we have organs that play vital functions, such as the beating of our heart. These organs contain proteins that play important roles, such as this ion channel that allows ions to cross in and out of the cell. Proteins are made of sequences of amino acids. Atoms in these amino acids interact with other atoms, and that allow the protein to have a three-dimensional structure and play a certain role within the body. Hydrogen and deuterium are pretty similar, each having one proton and one electron, though deuterium has a neutron as well. This makes deuterium heavier. But deuterium interacts the same way that hydrogen does in most situations. We can combine it with things like water to form what we like to call heavy water. We can use this in labeling our protein for a hydrogen-deuterium exchange. We can put our protein or peptide region in a deuterated solvent, and hydrogen and deuterium will spontaneously exchange, thus labeling our protein in its backbone. We can use this in an experiment where we take our peptide or protein of interest and put it in a deuterated solvent. Over time, more and more of the backbone will exchange, first the most accessible regions, and then, given enough time, the whole protein. This only happens if the hydrogen already isn't busy. We see here that the most accessible hydrogens exchange, then most of the hydrogens exchange, but we don't see the ones that are already interacting with the neighbors, the ones that are buried, or the ones that are very rigid. We can stop this exchange in its tracks by quenching the protein, lowering the pH somewhere between lemon juice and vinegar, and chilling the protein down so that the protein is stuck in its deuterated position, or frozen in time, like this protein here. We can quench our protein at different time points of our experiment and then give the protein a protease that digests the protein into small regions, like we call these peptides. And notice how more and more of these peptides are deuterated over time. So more and more exchanges happened over time. We can take our digested protein at a certain time point and run it through mass spectrometry. First, we sort out these peptides, give them positive charges, and run them through a machine, something like this, that accelerates our ions through a voltage and sorts the peptides by their size. Then we can analyze our data and see what happens. Here's a tracing of what we might see from a peptide that has not been deuterated. Now we can look at this peptide region over different time points. First at the very beginning, then in the middle, and then at the end of our experiment. Look at what happens to our spectrum. We see a shift more and more to the right. Our peptide region became more and more massive. Remember that deuterium is more massive than hydrogen. We can plot this difference, looking at the shifts in our peaks over time. And we know how heavy deuterium is, thus we know how much deuterium was exchanged for hydrogen. Here we see a big increase up to 10 minutes, and then our deuterium exchange leveled off from 10 to 100 minutes. And we can learn more about the dynamics of the protein through this. Take this linker region here, this loop that's pretty accessible to the solvent. This is what a plot might look like, where we have a lot of deuterium exchange right in the beginning when our peptide was most accessible to the solvent, and then it leveled off over time because most of the deuterium was already exchanged. Instead, let's look at this more internal region. Now we see not much deuterium exchange happening. This is because this is a really rigid, structured region of our protein. Thus, we don't expect a lot of the hydrogen to exchange with the deuterium because those protons are already busy. But we can change that. So take our loop region here again, the one that was really flexible and really accessible. Say we know a drug that interacts with this protein. We don't know where it interacts, but we're about to find out. We give the protein this drug, and look, now we see a flattening of our curve. This might tell us that this drug interacts either directly or indirectly to make that loop region less accessible to the solvent, more rigid, less likely to exchange with deuterium. So what did we learn from this experiment? We learned that deuterium is a heavier version of hydrogen, and we can use this to label our protein and analyze the dynamics of specific regions of our peptides. We use this by putting it through mass spec and looking at our data and seeing how heavier certain peptide regions got or didn't get. From here, we can learn more about flexible loops of ion channels and how these proteins act within our organ systems. We can learn about protein-protein interactions this way, protein-drug interactions, protein-toxin interactions, protein-enzyme interactions. And from here we can learn more about the body, learn more about health and disease, and learn more about science. I hope you enjoyed this Minute Biophysics video on mass spectrometry with hydrogen deuterium exchange. Thank you.